Hello everyone. Let's start with a new episode of cholesterol and its significance. So in this presentation we are going to cover some information regarding the cholesterol. What is cholesterol? What is its chemical property? Moreover, from where this cholesterol it has come, or how this cholesterol can be synthesized in the human body. What is the significant role of cholesterol in the human body, how this cholesterol it helps to build up the human body. Moreover, its synthesis, how the body it synthesizes the cholesterol. Moreover, its sources, from where the body it can get cholesterol. So keeping all this aspect, we are going to make out this presentation. So let's start without wasting any more second of time. So basically, you see the word cholesterol, it has derived from an ancient Greek word. There are two words you see here. This chole, it means the bile. Moreover, the stereos, it means the solid, right? Apart from that, if you see, followed by that, here we already got the chole, right? This is the chole. And from stereo we get this steer, right? Steer. Now we got this two word. Now there is one more term, this ol. Now this ol, where it has come. So you see here it is followed by a suffix ol, which is actually nothing but it's a alcohol. Thus you can say it's a organic molecule. Now basically this cholesterol, it is a lipid derived molecule, that is a sterol or a modified steroid. Its systematic name, you see here, it's a long name. Its systematic name is this. Moreover, it have for the cholesterol alcohol, it has this long big name, right? Now if we see again this cholesterol, it is the major sterol in the animal tissue. Most commonly found sterol in the animal tissue, it is the cholesterol, first of all. Moreover, you see here, this cholesterol, it is present in the tissue and in the plasma. But how it is present? We know that it is present in our human body, but how it is present? So it can either, this cholesterol can either remain as a free cholesterol, or it can remain in the storage form, which is actually it is combined with a long chain fatty acid. When this cholesterol is combined with a long chain fatty acid, it is known as cholesterol ester. I repeat, when the cholesterol it remain in close contact or it form a bond with a long chain fatty acid, it forms cholesterol ester, right? Clear? Now in plasma, in the human blood, in the plasma, both the forms, both this cholesterol, it's the free form plus the bound form, that is the cholesterol ester, both these forms are generally transported in the lipoprotein. So I hope what is the lipoprotein, you know. Lipoprotein, it is nothing but it's a combination of lipids and protein, right? So in the plasma, both these form, the cholesterol as well as the cholesterol ester, it is transported in the lipoproteins. Now plasma low density lipoprotein, LDL. In the plasma we have basically two types of lipoprotein. One it is LDL and one it is HDL. LDL stands for low density lipoprotein, HDL stands for high density lipoprotein. So now in the plasma, this low density lipoprotein, it is the vehicle. Actually the cholesterol use this LDL, right, to go from one place to another. That is in the plasma low density lipoprotein, it is the vehicle of uptake of the cholesterol as well as the combined form, that is your cholesterol ester, into many tissue. Thus you can say this LDL, it is the vehicle which is responsible to move the cholesterol in your body, right? Now free cholesterol, it is removed. I repeat, the free cholesterol, whatever it is present in your body, it is removed from the tissue or the plasma by high density lipoprotein. Thus you can say that HDL, it helps in removing or washing away of the cholesterol from the body. LDL, it helps in the transport of the cholesterol, whereas HDL, it helps in the removal. And once this cholesterol, it is removed from the tissue or from the plasma, it is directly sent to the liver, and from where the liver, it actually used to eliminate the cholesterol either unchanged. Sometimes it change means it sent out the cholesterol without performing any biochemical reaction over it, or sometimes this cholesterol, it get converted into bile acid. I repeat, in the liver, this cholesterol, once it is transported by the HDL, it get converted into the bile acid, and this process, it is generally known as the reverse cholesterol transport. I repeat, the conversion of cholesterol into bile acid, it is known as reverse cholesterol transport. Moreover, this cholesterol, it is the most abundant sterol in the human. I repeat this line, you should know this thing, that cholesterol, it is the most abundant form of the sterol which is found in the human. Again, if we see, this cholesterol, actually it is the major constituent of the plasma membrane and of plasma lipoprotein. So you should know this thing, this cholesterol, it is the major constituent of the plasma membrane of the cell as well as the plasma lipoprotein. Apart from that, we have already covered that this cholesterol, it is the precursor of bile salts. So I hope you know the use of bile salts. These bile salts are actually available in our body. They generally helps in the emulsification of fats, or you can say digesting fats, right, in the human body. Whatever the fat you used to consume, these are generally digested with the help of this bile. Now, it is a precursor, this cholesterol, it is a precursor of various steroid hormones, which also includes the adrenocortical hormones, right? Apart from that, hormones, sex hormones, placental hormones, etc., these are actually formed from this cholesterol only. Moreover, this cholesterol, it is also a precursor of vitamin D in the human body. Now, this cholesterol, apart from this, they also helps in the nerve transmission, sending the signal of the nerve from one point to another. Actually, this cholesterol, it used to help. Moreover, this cholesterol, it is the major constituent of the gallstone. So sometimes we used to heard that person has suffered from gallstone. Gallstone is the formation of stones, right, in the gallbladder. So these gallstones, these are actually formed by this cholesterol only. Moreover, this cholesterol, it has a main role in the pathologic process as a factor of genesis of atherosclerosis. It's a vital artery disease. Means with the passage of the time, you can see the patient or the person who used to have high cholesterol diet, they suffer from cardiac blockage or arterial blockage. So this is generally known as the atherosclerosis. Actually, it is the formation of plaque in the artery. It is a condition where the plaque, it forms in the artery, which actually prevents the flow of the blood to the heart, which sometimes may lead to the failure of the heart, right? Now, sources of cholesterol. So from where we used to get the cholesterol in our body? First of all, from the diet, right? Next, de novo synthesis. So we have already covered the fatty acid synthesis. So de novo means the synthesis of the cholesterol that can also happen, or that can be done within the body itself, right? Apart from that, hydrolysis of cholesterol ester. So we already discussed what is cholesterol ester. It's a combination of the cholesterol along with the long chain fatty acid. So if we hydrolyze this, because this is a ester, right? So acid and alcohol. Earlier we know that the cholesterol, it is a derived alcohol, right? It is having a alcohol-like property, as well as when this alcohol, it will combine with a long chain fatty acid, that's a acid. So acid and alcohol, when they will combine, they will form the ester, right? Which a elimination of a water molecule. So if we hydrolyze, if we add one molecule of water to that, this bond of the ester bond will break, and you will finally get the cholesterol or the alcohol separate as a separate entity. Moreover, you will get the long chain fatty acid as a separate entity, right? Now you should know that more than half of the cholesterol in the human body, actually it basically arises by the synthesis. The body used to synthesize the cholesterol, and the remaining part, or the remainder volume, apart from that, it is coming, or it comes from the average diet, whatever you used to take. So most of the cholesterol, you remember, most of the cholesterol in the human body, it is generally synthesized by the body itself, right? Now the liver and the intestine, they actually accounts for approximately 10% each of the total synthesis in the human. So these are the two primary source of genesis of the cholesterol, the liver and the intestine, and they account for around 10% of cholesterol synthesis. Apart from that, all the tissue which contains nucleated cells, means a cell with a nucleus. So all nucleated cells in your body which are present in different tissue, they are capable of cholesterol synthesis. So I repeat again, all the cells in your body, but the condition is that the cells should be nucleated one, not the RBC, right? So all the nucleated cells in your body have the property, or has the capability to synthesize this cholesterol, and this synthesis actually it occurs in the endoplasmic reticulum and in the cytoplasm or the cytosol. The synthesis of the cholesterol, it used to occurs in the endoplasmic reticulum and in the cytosol. So function of the cholesterol. So function of the cholesterol, first of all, cholesterol, it is required to build and maintain the membranes, basically the cell membranes, right? It is an important constituent of the cell membrane. Now, because of which actually this cholesterol, it modulates or it increases the membrane fluidity of the cell, right, over a range of physiological temperature. Because of this cholesterol, I repeat, because of this cholesterol only, the fluidity of the cell membrane increases over a wide range of temperature in your body. Moreover, this cholesterol, it serves as a precursor, right, for the biosynthesis of steroid hormone, which we have already discussed regarding the adrenocorticoids, right? It also acts as a precursor for synthesis of the bile acid as well as the vitamin D. Now this cholesterol, it is the principal sterol synthesized by all the animals, right? So you should know that all kinds of the cells in the animals, they can produce or they have the property to synthesize this cholesterol. Moreover, you see, as per the recent study that has been done or performed in the human, it has been found that a vitamin D, right? Cholesterol is a precursor of vitamin D. We know that, right? Now, this vitamin D is a potent antioxidant. So we know how a antioxidant, it prevents our body from the free radical mechanism. So once this free radical it forms in our bodies, this free radical can lead to different destructive process in our body, like aging, right? Sometimes disease, pathological condition like cancer, it also occurs due to the free radical only. But this vitamin D, it acts as a potent antioxidant. They have the capacity to combat the free radical. Thus, you can say this vitamin D, which is a means cholesterol, right? From the cholesterol only you get the vitamin D, and vitamin D has the property to detox your body, to detoxify your body, right? Moreover, in the cell membrane integrity, it generally helps in the cell membrane integrity, which we have already discussed. That is, it is actually biosynthesized by the all animal cells, right? We have already discussed. And because it is an essential structural component of the animal cell membrane. This cholesterol is a important structure of animal cell membrane, and it is actually required to maintain the structural integrity and fluidity of the cell membrane. I repeat, this cholesterol actually it helps to maintain the structural integrity and fluidity of the cell membrane. Now, if we see how the cholesterol is synthesized in our body, so basically the synthesis of the cholesterol takes place in 5 different steps. So if we see the steps, the major steps are, first of all, the biosynthesis of mevalonate occurs from acetyl coenzyme A. That is the first step, right? The next step, it is the formation of isoprenoid units from the mevalonate by loss of carbon dioxide. That is the second reaction or part of the reaction, you could say. The third part, it is the condensation of 6 isoprenoid units to form a squalene ring, right? The fourth step, it is the cyclization. It will now form a cyclic structure. Cyclization of a squalene give rise to the parent steroid which is known as lanosterol. Lanosterol, it is the parent steroid, right? And formation of cholesterol from the lanosterol, that is the last step. So I repeat once again, from the acetyl coenzyme A, first the mevalonate will be formed with the loss of carbon dioxide, right? Next is the condensation, from the acetyl coenzyme A, first you will get the mevalonate, right? From the actually mevalonate you will get the isoprenoid. Afterwards, from the isoprenoid you will get the squalene. From the squalene you will get the lanosterol. From the lanosterol we are going to get the cholesterol. So these are the 5 basic reactions which takes place in your body, and regarding which we are going to discuss in our next slide. Okay, so now we are going to start with the steps. So which are the steps, what are responsible for the synthesis of the cholesterol? So the first step, as I already told you, it's the biosynthesis of mevalonate. So from where we are going to get the mevalonate? So let's start. The first step of the synthesis, it is the cholesterol synthesis, it is the formation of mevalonate from the acetyl coenzyme A. This is the initial or the first product from where it is going to be made. So you see here, now this reaction takes place in the cytosol of the cell. For preparation, it is always the cytoplasm, the site for the preparation of cholesterol. Now the next step, it is what actually happens. Initially you see here, initially 2 molecules of acetyl coenzyme A take condenses, right? They condenses in the presence of the enzyme thiolase to form acetoacetyl coenzyme A, right? The next step, now in the next step, what actually happens, you see here, the acetoacetyl coenzyme A here, it condenses with a further molecules of acetyl coenzyme A, and this reaction, it is actually catalyzed by a special enzyme. This is the HMG-CoA, means actually it is the beta methyl glutaryl coenzyme A. This is the HMG-CoA synthesis. Actually this enzyme, this synthase, HMG-CoA synthase, it is responsible for addition of another molecule of acetyl coenzyme A to the acetoacetyl coenzyme A, which results in the formation of the 3-methyl glutaryl coenzyme A, which is also known as HMG coenzyme A. And this reaction, actually it is here it is actually reduced to the mevalonate actually by the NADPH. Here you can see. Now what actually happens here, the acetoacetyl coenzyme A, it actually condenses with a further molecules of a acetyl coenzyme A. This is catalyzed by HMG coenzyme A synthase to form HMG coenzyme A, and that is reduced to mevalonate. Here you see it is reduced to mevalonate, and by the NADPH, you see 2 molecules of NADPH, these are involved here, right? So by NADPH, and it is also catalyzed by a enzyme which is known as the 3-hydroxy 3-methyl coenzyme A reductase. It is also known as the HMG CoA reductase enzyme here, right? Now in the next step, what actually happened, you see here the synthesis of the mevalonate. It is the principal regulatory step, or you can say it is the crucial or rate limiting step for the synthesis of the cholesterol in the human body, right? So this step, it is actually this particular step where this enzyme HMG CoA reductase, it actually appears, or it actually works. This is the site for most of the drug which are generally used to lower the cholesterol in the human body. So if a person, most of the person who are generally obese, who have high amount of the cholesterol in their body, so the physician, they used to have, or they used to give this particular classes of the drug which actually act on this particular step. So it is the site for most of the drug which are used to lower the cholesterol in the human body. For example, I have given here the statin drugs, right? These statin drugs are generally used to lower the cholesterol in the obese patient, right? Now moving to the next step. The next step, it is the formation of isoprenoid units. So how it is going to form, you see here. First of all, the mevalonate, it has formed. We have come to know that 2 molecules of acetyl coenzyme A, they first condenses, then we get the acetyl coenzyme A, then again we get the mevalonate. Now from the mevalonate, now what is the fate of the reaction? Now this mevalonate, it is phosphorylated sequentially by ATP. ATP will phosphorylate it by 3 specific kinase. So the reaction, it starts here. You see, it is the mevalonate, right? So first kinase, it is the mevalonate kinase. Here ATP, it is working, right, to form the mevalonate 5-phosphate. 1 phosphorus group has been introduced from this ATP, right? From this, the next phosphorus group will be again added. Here the phosphomevalonate kinase, the second kinase enzyme. I already told you 3 kinase enzyme. Again 2 phosphorus group. Here again we get the diphosphomevalonate kinase, and in all these 3 steps you can see here ATP, it has been involved, and these are the 3 kinases: mevalonate kinase, phosphomevalonate kinase, and diphosphomevalonate kinase, right? So these are the catalyzing agent for this conversion, right? Next, afterwards, actually what happens, generally decarboxylation takes place here. So generally diphosphomevalonate decarboxylase enzyme works on this particular product which has been formed to form the final product that is known as the isopentyl diphosphate, right? It is the isopentyl diphosphate unit will be formed. Now what actually happened, condensation of these 6 isoprenoid units to form squalene. Now how this condensation it will take place, I again go to the previous slide. You see here isopentyl diphosphate. So how many carbon? 1 2 3 4 5 means you are having 5 carbon here, right? You are having 5 carbon here, which is acting as a isoprenoid units, right? Now these are going to get condensed. So how they are going to condense? So in the next slide you see condensation of 6 isoprenoid units. It is because the squalene it is a 30 carbon atom. So as we know that the isoprenoid, it have only how many carbon atom? 5 carbon atoms. So therefore we need to have 6 isoprenoid unit to get condensed. 6 isoprenoid unit of 5 carbon. The isoprenoid unit have 5 carbon. Therefore 6 isoprenoid unit, they are going to condense to form the squalene, right? So this is the basic sequence. You see, for first C5. So we are starting with the C5, means that is the simple isoprenoid unit, then it will get converted into C10, then it will get converted into C15, and finally it will get converted into C30. So this is the sequence of formation of the cholesterol. So we are going to start with the C5, then we are going to get the C10, then C15. So gradually the chain will elongate in this way, right? Now how this condensation it takes place. So we are again going to start the reaction. So basically you see here, you just see in the given chart or in the reaction, the isopentyl diphosphate, that is the isoprenoid unit, right? It will be now isomerized by a shift of a double bond to form dimethyl allyl diphosphate. So this is the isopentyl diphosphate. Now it will get isomerized to form dimethyl allyl diphosphate. It is again then condensed with another molecule of isopentyl diphosphate to form a 10 carbon. I repeat, a 10 carbon C10 carbon intermediate. This intermediate it is known as the geranyl or geranyl diphosphate. Done. So here we are going to get the geranyl diphosphate. From this geranyl diphosphate, the reaction will again proceed. You can see the reaction here. So we are starting with the isopentyl diphosphate. It will get isomerized. Then we are going to get the geranyl diphosphate, right? Now moving to the next part. Again, what will happen here? Actually, see, a further condensation will take place. As I have already told you that C5 to C10 to C15 to C30. Now, a further condensation will take place with isopentyl diphosphate to form farnesyl diphosphate. So here again, you see here isopentyl diphosphate. It has finally formed the geranyl diphosphate. It's a C10 carbon, right? Again, condensation will take place. Again, condensation will take place to get a C15 compound. This is known as farnesyl diphosphate, right? Now from here, what actually happened? Two molecules of farnesyl diphosphate, as it is C15, right? So two molecules of farnesyl diphosphate, they will condense together at the diphosphate end to form a squalene. The squalene is a C30 compound, means it is having 30 number of carbon, right? So initially the inorganic pyrophosphate is eliminated in this reaction, forming a pre-squalene diphosphate, which is then reduced by NADPH. So NADPH will act as a reducing equivalent here with the elimination of a further inorganic pyrophosphate molecule in the synthesis of the cholesterol, right? Now the next step, it is the formation of lanosterol. So from this squalene, lanosterol, it is going to get formed. So how they are going to get formed? You can see here. The first step is that the squalene can fall into a structure that closely resembles the steroid nucleus, right? This squalene can form a structure which is similar to that of your steroid nucleus. So the reaction you see, this is the squalene, right? So these are the number which are given to the defined carbons, right? So here it is important to note the number of the carbon 13, carbon 14, carbon 8. So what actually happens here? Actually, you see here, before this ring closure, once this ring has formed, now it is going to close. So before this ring closure, squalene is generally converted into squalene 2,3 epoxide. So this is the squalene epoxide compound, 2,3 epoxide by a mixed function of oxidase. That is, here what enzyme will act? It is the squalene epoxidase. So epoxidase in the endoplasmic reticulum. So generally these steps it takes place in the endoplasmic reticulum by virtue of the enzyme squalene epoxidase. Now in the next step, the formation of lanosterol, you see here what actually happens. Again, the methyl group, I already told you to observe the carbon at the position 13 and 14. What actually happens? You see the methyl group on the carbon 14, this methyl group, now it will be transferred. You can see here with a dotted line, this will be transferred to the carbon 13. The methyl group which is present in the carbon 14 will be transferred to the carbon 13, and the other carbon which is present, and the other methyl group which is present on the carbon 8, this one, this methyl group, now it will be adjusted to the carbon 14, means this carbon will shift to the carbon 13, and from the 8 position it will again shift to this position. So this process, it is catalyzed by another classes of the enzyme which is known as oxidosqualene or lanosterol cyclase. So these steps, it is catalyzed by this particular enzyme. So ultimately this will lead to the cyclization, means the closure of the ring, and this newly formed cyclic structure. Now once it will be transferred to from this position to here, the next methyl group from here to here, now the ring is going to close. The ring is going to close. So once this ring will close, it will take up the structure of the lanosterol, right? So in the next step, the formation of cholesterol. So how it is going to form? So the last product, that is the formation of cholesterol from the lanosterol, it takes place in the membrane of the endoplasmic reticulum. You should remember this conversion of lanosterol to the cholesterol, it always takes place in the endoplasmic reticulum, and it involves the changes in the steroid nucleus. So this is the steroidal nucleus. There are some changes will take place. So what are the changes it is going to take place? So these are the basic steps you see. From the lanosterol, it has converted into 1,4-desmethyl lanosterol, then zymosterol isomerizes to cholesterol, then to desmosterol, then finally it is going to form the cholesterol, right? So the changes here to note, the first changes is here the methyl group on the C14, right? You remember the methyl group on the C14 and C4, now they are removed to form 14-desmethyl, means 2 methyl group has been removed. The first step, the first carbon on the the methyl group on the C14 and the other one in the C4. If you want to see the structure, I am again going to show you the structure here, just see in the previous slide. So the methyl group which is present in the C14, other one it is the fourth carbon, right? So these two are going to now get converted or interchanged, right? Now they are going to convert it, or they are going to remove to lead to the formation of 14,1,4-desmethyl lanosterol, right? And then zymosterol. The next step, it is the zymosterol. Again, if you see in the structure, the double bond which is present actually here in the double bond which is present between the C8 and C9 position is subsequently moved to the, here I am showing you in the red arrow, you see here the double bond will be moved to the C5 and C6 in the 2 steps, forming a desmosterol. Here the double bond will be shifted, and it will lead to the formation of the desmosterol. From the zymosterol isomerizes to form the cholesterol, and from here it is going to shifted, this double bond, it is going to get shifted to finally form the desmosterol, right? And finally this is led to the, finally the double bond or the side chain, it is reduced, right? The double bond will be reduced to produce the cholesterol. So this is the synthesis of the cholesterol. So thank you everyone for your patience hearing. I hope this slide make you understand the concept of the cholesterol, significance of the cholesterol, how it is synthesized in the body. If you like my video, just press the like icon. You can share this video with your colleagues and your classmates, and for further notification from this channel you can press the bell icon. Thank you everyone.